Well, my Bluetooth still isn't working. I don't know what's up with this thing, but it just doesn't want to connect. Or maybe my computer has something personal against it. I don't know. But it's not connecting, so I'm gonna stop wasting my time with it. All right. Okay, Ketogenesis. Let's start with a word of prayer and then we'll dive in. Father, thank you for this day. It's cold, but thank you. It's our last day of instruction. Escort us now into finals week. Watch over us, please. Give us the fortitude to go to the finish line, cross it, and feel very good about the work we've done. And reward us, Lord, for being faithful, even when things are really not all that opportune. In Jesus' name we pray. Amen. All right. So during periods of time when you have high rates of fatty acid oxidation, this is particularly the case in the liver, you have very large amounts of acetyl coenzyme A that are generated. And the main source of acetyl coenzyme A is definitely the oxidation of fatty acids. Amino acid turnover does account for a small percentage of that turnover, about 5%. So to give you an idea of oh dear. There we go. We're looking for my blackboard here. So if we take, for example, it's usually the branched chain amino acids. So this includes leucine and valine and iso. Good gravy. Focus, you. And isoleucine. I'll turn the lights on. That's a little better. These are actually oxidized in the mitochondria. The only other amino acid that's oxidized in the mitochondrion in animals, I must emphasize that in animals, is glycine. It's not the case in plants, but it is in animals. So with leucine, for example, so all the different branch chain amino acids are going to first undergo deamination. And in this case, you're going to have alpha keto glutarate, of which there's plenty in the mitochondrion, will pick up that amino group and it will make glutamate. And this is a branched chain amino acid transaminase. This is a enzyme that essentially will work just fine for all three amino acids. When you do that, you make something called alpha keto isocaproate, and it's just leucine essentially minus the amino group. So it's beta keto 1, 2, 3, 4, 5 iso caproate 1, 2, 3, 4, 5. So it's caproate. So it's beta keto isocaproate. This is then going to go through a so-called branched chain alpha keto acid dehydrogenase. So this is going to make an acetyl coenzyme A group out of this. It's going to do something very similar to what alpha keto glutarate dehydrogenase or pyruvate dehydrogenase do. So you'll have free coenzyme A plus NAD. Those are then going to give you reduced plus a hydrogen ion plus CO2. So you get an oxidative decarboxylation, essentially, just like the other reactions we're so very familiar with. And you're going to get a beta isocaproyl coenzyme A. Sorry, it's not caproyl, it'd be butyryl, because this takes a hike and then, so this would be isobutyryl coenzyme A. The branch chain alpha keto acid dehydrogenase will handle leucine and valine and isoleucine derivatives. And mutations in structural genes that encode this enzyme will lead to a buildup of these alpha keto acids. Those alpha keto acids will then be broken down by oxidation to particular things that are released in the urine. And those things that are released in the urine kind of make the urine thick. And because it's thick, mutations in this will cause a genetic disease called maple syrup urine disease. I don't name them, I just report them. I would never think of naming a genetic disease this. Maple syrup urine disease. And maple syrup urine disease causes disease, it's a genetic disease that causes death rather early in life. Okay, getting back to what goes on here, you then essentially are going to have several steps. And so about 3 more steps. This will eventually give you a HMG coenzyme A. HMG coenzyme A stands for beta hydroxy beta methylglutaril coenzyme A, and then there's an HMG coenzyme A lyase. And that HMG CoA lyase is going to give you acetoacetate plus coenzyme A. So there's some coenzyme A that can contribute to the formation of ketone bodies, but it's not a lot. It's only about 5%. This, of course, is a ketone body as well, acetoacetate. So leucine degradation can contribute to ketogenesis. All right, so the excess acetyl coenzyme A will exceed the capacity of the citric acid cycle. And so you get spillover pathways, and these spillover pathways include the synthesis of ketone bodies. So this is simply the result of the citric acid cycle not being able to keep up. So ketogenesis occurs in the mitochondrion. It consists of the synthesis of 3 different molecules: acetone, acetoacetate, and D-beta hydroxybutyrate. These 3 chemicals are called ketone bodies. This process whereby they are synthesized from excessive coenzyme A is called ketogenesis. Now, of these 3 molecules, acetone is volatile and is therefore exhaled. And a starving person may, at least at the first, have a musty breath about them, and that musty breath is acetone. After a while, acetone synthesis, because it's a carbon sink, will decrease and the musty breath will go away. These other 2 molecules, acetoacetate and the beta hydroxybutyrate, are transported through the blood and are used as energy sources for tissues during the post-absorptive state. Ketogenesis is enhanced by the hormone glucagon. It is, however, inhibited by the hormone insulin. So the brain tends to want to use glucose. During periods of starvation, the brain can use ketone bodies. It doesn't prefer ketone bodies, but it can adapt to ketone bodies. So typically, if someone starts to starve, there's usually a period during which their thought processes will fog. There'll be some mental fogging that will tend to go away as their brain starts to adapt to the use of ketone bodies for energy. So the first step involves the synthesis of acetoacetyl coenzyme A from 2 molecules of acetyl-coenzyme A. This reaction should look really familiar. In fact, this is the last step of beta oxidation, except it's in reverse. And this is catalyzed by thiolase, except it's a different thiolase than the one that is used for beta oxidation. So humans express 2 main thiolase genes. One is for ketogenesis, the other is for beta oxidation. And so the thiolase gene, ACAT1, is used, that the enzyme encodes by the gene ACAT1, that's used for beta oxidation. The ACAA2, that's used for ketogenesis. You know what? I mixed those up. I'm sorry. ACAT1 is a gene for ketogenesis. The ACAA2, also called HEDHB, either one of those, that's the beta oxidation gene. So, oh goodness, let's see here. All right, so if you have so you get 2 molecules of acetyl coenzyme A. Those are going to, and you'll liberate a free coenzyme A. These are high energy bonds. You don't need to put any energy into this, and you're going to make so essentially this bond gets sent over there, that hydrogen gets sent there, and you get a free coenzyme A. All right. So this is catalyzed by thiolase. ACAT1. All right. The next step I was going to add another acetyl coenzyme A to this molecule here. So the acetoacetyl coenzyme A. Now add yet another acetyl coenzyme A. You're also going to need a water to get this reaction to go. And you're going to make beta-hydroxy beta-methylglutaryl coenzyme A, which is mercifully abbreviated HMG coenzyme A. That's step 2. Now, HMG coenzyme A is an intermediate in the synthesis of cholesterol. So with this reaction, you're going to add an acetyl coenzyme A, that's a thioester, plus water. You'll liberate a free coenzyme A this way. This will be HMG CoA synthetase. Now, and this will give you oh, do I have that right? 1, 2, 3. Yeah, okay. And this is beta-hydroxy beta-methyl glutaryl coenzyme A. Again, HMG coenzyme A. Now, word about this enzyme here. In the cytoplasm, you have HMG CS1. This is the enzyme involved in cholesterol biosynthesis. This enzyme is cytosolic. The enzyme, however, for ketogenesis is HMGCS2. This is used for ketogenesis. This is mitochondrial, and it's used for ketogenesis. So yes, there's two versions of this enzyme. They catalyze the same reaction, but they're involved in the production of different products. Okay, so remember what we did with leucine. We made this particular product with leucine, and we busted it apart. Going back and forth just like a yo-yo. Now we're going to bust it apart with HMG, beta-hydroxy beta-methylglutaryl coenzyme A lyase. So we will take our molecule and we will convert it to acetoacetate and acetyl coenzyme A. This very enzyme is the one that is used in the degradation of leucine. So once again, back to the blackboard. I'm going to keep my carbon straight here. I'll add an extra carbon. Yeah, okay. Okay, this is HMG coenzyme A. Now, we're going to subject it to HMG-CoA lyase. And we're going to get from this coenzyme A plus, we're going to get so essentially this piece here gives rise to that molecule. Then this bit over here that's going to give rise to our acetoacetate. Now, this acetyl coenzyme A, that can just be recycled. So this is going to be recycled for more ketogenesis. This up here is now your starting material for the synthesis of your other ketone bodies. So if you decarboxylate that, which is actually not that hard to do, and that's acetoacetate decarboxylase, that is going to generate something very familiar to all of you. And if you don't recognize that molecule, you should have a chemistry set thrown at you. This, of course, is acetone. And then the other reaction, and this reaction is reversible, will essentially take reductant and you could just as easily reverse this reaction. And this is D-beta hydroxybutyrate dehydrogenase. And that is simply going to give you a reduced, oh, good gravy, that's a carbon. So eager to draw oxygens. So now instead of the ketone here, we have ye holy alcohol, and that is beta hydroxybutyrate. 1, 2, 3, 4, beta hydroxybutyrate. And so this molecule and this molecule and this molecule, those are your ketone bodies. And this is the basis of ketogenesis. Mutations, by the way, in the HMG coenzyme A lyase will result in something called you can't break down the beta hydroxy beta methyl glutarate coenzyme A, so you get a HMG aciduria. HMG aciduria if there are mutations in that. This is treatable by diet, by restricting amino acid leucine, because remember, this is also involved in leucine degradation. If you don't address this early in life, the patient dies quite early due to the aciduria. Okay, so we get decarboxylation from acetone. That acetoacetate decarboxylase, I must tell you, is subject to downregulation. That downregulation is simply the result of losing carbon through the lungs. So as ketogenesis continues, the larger amounts of acetoacetate and the beta hydroxy, D-beta hydroxybutyrate, those larger amounts will start to shut down acetoacetate decarboxylase. Therefore, the carbon sink is ended. The carbon sink is ended. And here's this is just shamelessly stolen from your book, giving you the ketone bodies right here. So here, here, here. Those are your ketone bodies. Ketone bodies are then, when they're made in the mitochondrion, they are moved into the blood by means of so-called monocarboxylate transporters. There's a MCT1 and MCT2 that will kick them into the blood from the hepatocyte, and these will flow through the blood and will be able to feed your tissues. And these other tissues will use ketone bodies as fuels. So the beta hydroxybutyrate can be used to make reductant. You can convert it to acetoacetate. Then the acetoacetate is just oxidized by thiolase into coenzyme A. So it is essentially fuel for the Krebs cycle. That's why it's a good energy source for tissues that are subject to starvation when the blood glucose level goes down so low that uptake by facilitated diffusion is no longer efficient. And so you can see the interconversion of beta hydroxybutyrate to acetoacetate to acetoacetyl coenzyme A, and then back to acetyl coenzyme A. And this enzyme here, the beta keto acyl coenzyme A transferase, prevents you from burning 2-ATP to put a coenzyme A group on this. You simply take your succinyl coenzyme A, of which there's plenty in the mitochondrion, and since some of the succinyl coenzyme A is diverted for biosynthetic purposes anyway, you can divert some of that to make acetoacetyl coenzyme A. Then you just make succinate. Succinate just goes straight back into the Krebs cycle. And then you make two acetoacetyl coenzyme A and then two acetyl coenzyme A's, and you feed it back in the Krebs cycle. Which is told here. I said that already. You saw it on the slides again. Acetoacetyl coenzyme A gets converted back into 2-acetyl coenzyme A's, and away you go. Now, during starvation and also in people with poorly controlled diabetes mellitus, because you have low insulin and high glucagon levels, ketogenesis starts to run away, and you get overproduction of ketone bodies. Because you don't have sufficient insulin, in the case of type 1 diabetes mellitus, or because your body doesn't respond properly to insulin, you have insulin resistance, as in the case of type 2 diabetes mellitus, your body can't depend on glucose for a carbon and energy source, so it turns to fatty acids. So you oxidize fatty acids, you generate far more acetyl coenzyme A than your body can handle, and your cells can double, than the Krebs cycle rather can handle. Therefore, you will divert those into the synthesis of ketone bodies, because glucagon also upregulates the enzymes of ketogenesis. This is called ketosis. You get mild ketosis when you don't eat. So for example, this summer, when I went in for my physical, it was in the afternoon, so I said, oh goodness, it's too nice a day to not do anything, so I went for a bike ride. And because you have your lipid panel, you have to not eat for 12 hours, so I got up in the morning, I did the bike ride, and then I went and had the physical. The physical, I think, was at 1 o'clock. And when I got my blood work back, I had some ketone bodies in my urine. And It wasn't very much, but the doctor said, oh, that's just your bike ride. That's, you didn't eat, so your body turned to stored fat and made some ketone bodies, and they got kicked into the urine. Now, in someone with uncontrolled diabetes mellitus, the levels of malonyl coenzyme A will increase such that fatty acids can't be admitted into the mitochondrion for oxidation. Because you are going to make malonyl coenzyme A, you'll make lots and lots of ketone bodies, but you can't bring glucose in properly for glycolysis, and you can't properly oxidize fatty acids because of high levels of malonyl coenzyme A. So essentially, your cells start to starve. And this is potentially a very dangerous situation because now your blood starts to run thick with ketone bodies. Those ketone bodies are organic acids. They drop the pH of the blood, and that is called acidosis, but because it's due to ketone bodies, it's called ketoacidosis. These ketone bodies also act as diuretics in the kidney. In order to excrete them, your kidney has to make sure that there's plenty of water to keep them soluble. So you reabsorb less water. So you have acidosis, you have dehydration, and you can't feed your tissues. This is a very dangerous situation. Now, typically, if you don't have diabetes mellitus, these ketone bodies are used as an energy source while you're waiting to be able to get your next meal. Glucose is then made via gluconeogenesis, and that is exported to the brain as fuel. Glucose will be spared by other tissues because they will use these ketone bodies, so heart, skeletal muscle, kidney, and some aspects of the brain, but the other aspects of the brain will try to opt more for the glucose. And gluconeogenesis is induced by glucagon, and it's also induced by cortisol. Cortisol also induces muscle protein breakdown, and those amino acids will be the material for gluconeogenesis. And if you have trouble taking up, can't get enough glucose, they might also be fodder for ketogenesis as well, but fatty acid degradation will also be the material for ketogenesis as well. Okay, so untreated diabetes mellitus can cause severe dehydration, severe electrolyte imbalances, and that is potentially quite dangerous. So the best thing you can do if somebody starts to pass out due to some sort of diabetic coma, the best thing you can do is put some sugar under their tongue. And type 1 diabetics will typically have a kit with them, and inside that kit will usually be some goo. You just squirt it into their mouth. That'll give them enough sugar to be able to become conscious. They'll still feel like caca, but they can usually get up and do something. If things are really dire, they will have a little orange box, and that is a syringe of glucagon. And you jab the glucagon into the biggest muscle you can find, which will usually be the quadricep. That will cause a massive glycogen dump of all their liver glycogen, but it'll raise their blood sugar so they can become conscious, go get a meal again. They'll feel like caca times 10, but they will be conscious. And let me stop right there and ask our resident diabetes expert if she has anything to add to that.
No.
No, all perfectly clear. Oh, okay.
Mm-hmm.
Okay, well, then that's all I have to say, unless anyone has any questions.
Have you ever been in that situation, Amanda?
Amanda, did you hear the question?
No, could you repeat that? Which situation
Irma
specifically?
asked you if you had ever had that happen to you.
Which situation specifically are you talking about?
Diabetic coma.
No, luckily, it is very, very uncommon for type 1s. We tend to have we tend to develop a tolerance to fluctuating blood sugars. So like a blood sugar that could be really high or low for you, that might put you in a comatose, I can be, fine with. Our body tends to be a lot more tolerant to those fluctuations after a while.
And it depends on the diabetic. Some are more brittle than others.
Yeah, I think when I first was diagnosed, my blood sugar was 855.
Oh my.
Oh dear.
Yeah. I was supposed to be comatose, but I wasn't. So
No, I had an experience. Well, I didn't. My brother had some friends when he went to San Francisco State. North of San Francisco are the San Rafael Hills. I don't know if any of you had the privilege of actually being out there. That is some just drop dead beautiful country. But there's some great hiking there. And he had an acquaintance who had type 1 diabetes. And this gentleman, unfortunately, did not take terribly good care of himself. And he went hiking, and he did not take anything with him in the form of snacks or anything like that. And he started getting weak and started sweating and started talking funny. And so they put his hands over their shoulders, started to walk him in. Then he just plain old passed out. They didn't know what to do. So they took out his insulin and they gave him a shot. Now, his blood sugar was already too low. What does insulin do to blood sugar? Does it raise it or lower it?
Lower it.
Lowers it. So it took his already submarine blood pressure and lowered it even lower. It killed him. It killed him. Now, were those students well-intentioned?
Yeah.
Yep, they were well-intentioned. What killed that kid was ignorance. What killed him was ignorance. Now, granted, their buddies should have probably educated them a bit on, also he should have taken snacks with him. But that is one reason why science education is a bit of a passion of mine, because ignorance can literally kill people.
ignorance has been like one of the most frustrating things for me growing up, because so little diabetics are type 1. So like I remember even like in elementary school, like if people would like pass out candy or cupcakes or whatever, they would just like skip over me. And it always upset me so much. Or people would like, I had a lady once tell me that I just needed to eat more salad. And then like, yeah, it's insane. I've even had like ignorant doctors, like who have like, I had a doctor once who didn't know what IOB was, insulin on board. And it's insane.
Yeah. So that's why we are on a quest to educate everyone. And you should be too. Meredith, was that?
Yeah, I have a question about the internet assignment. On assignment 3, question 1, in the email you sent us, you wrote to use the human genomic transcript, but in parentheses, mouse G plus T. Is it-
Three, refresh my memory what three is.
Meredith, I think we used the mouse one, and that's what worked for us.
You're supposed to use the mouse.
on because there's one of them is on assignment 2 question 11, and that's the mouse one. But then I thought assignment 3 question 1 you said mouse. It said human on one spot and then mouse on another.
I have to look at it. I can't pull that out of my head. Meredith, 3?
Yes, assignment 3 question 1. Because I did it with the human one and it comes up with prostaglandin, I believe.
Yep, that's what I got.
Okay. So it's supposed to be human.
Yep.
Thank you.
Where does it say, Meredith, where?
So you sent us an email clarifying that one
Yeah,
on the 3rd, and so it says and choose the human genomic plus transcript, but then in parentheses mouse G plus.
no, it says mouse G, but that's the drop-down box. There's 2 drop-down boxes, mouse genome plus transcript, mouse G plus T, or human genome plus transcript, human G plus T. It's human genome. The human G plus T just means human genome plus transcript.
Right. I was just wondering because on the
That's
email,
all it means.
like one part of it said human, one part of it said mouse. So I just wanted to make sure I did.
No, it's a human
Okay.
genome transcript plus genome plus transcript. That's what you use.
Okay. Thank you.
And yeah, that worked. It worked quite well.
I have a question about assignment 3, question 5. So I get to the sequence, but then I'm not completely understanding what I'm supposed to be looking at to answer some of the questions.
3. Okay, so you compare those. Okay, so oops, no. All right. All right, so you come on, computer. All right, so you get to here and you do the comparison and it goes chugga chugga choo choo, and then okay, and there's your comparison, and it should give you, there's your identity right here and all that, and it asks you then to what's your identity, that's right there, blah blah blah. Then you get the nucleotide sequence, You get the nucleotide sequence simply by going you can essentially take a look at these. So that will give you the nucleotide sequence. So you could go to the gene and then get the nucleotide sequence. You then do the same thing, except you do it with the BLAST for nucleotide, and then you get an identity, and you compare that with the amino acid identity.
Okay, that
And
makes
then
sense.
you, so then you take it, what's percentage, which parts are well conserved, look at the sequence, where do you see the most? You'll notice the front of the protein's not very conserved. The back part is much more conserved. And then to find, that's what you do in Firefox, whatever you do in other servers. You do a find and you look for that sequence, and that's the one essentially that's modified by aspirin and blah, blah, blah.
Okay, so when I'm looking for the X's, so the X's are from the first search, right, with the amino acid code and not the, so that's on the page that you already have up, right?
Right, this, so you get, so when you, so this, see this plus here, Jess?
Yeah.
That's a conservative change. You have leucine, isoleucine. These two have the same biochemical properties. Lysine, arginine, they're both basic amino acids. That's a conservative change. So where you have like a stretch here, that doesn't have a lot of identity, but it has a lot of the same type of amino acids.
Okay.
Okay? And then you get through this portion here, now you start to get quite a bit of identity. And then up here, you notice there's gaps and there's just not a lot of conservation up here, whereas down here you get a lot more.
Okay, so when it says where one protein has several X's in a row, is it referring to, what is it referring to?
It's referring to places where you, oh, blast it. Yeah, the X's, they changed it so that there aren't so much X's as just spaces now. Where you have spaces, you don't
okay.
have similar changes here. You've got
Got it.
different sequence like there, and there, and there, and what have you.
Okay, that makes sense. Okay. And then for assignment 4, question 1, it says to construct a rough structure of the protein. I wasn't sure exactly what you wanted that to look like.
What's a rough structure mean?
That was what I didn't know.
What's a rough anything? What's a rough draft?
Before the finished product.
Then based on this, give me a rough estimate of what the protein probably looks like.
Okay, so it's not supposed to be one of the different images that's already on the website.
No, it's not. Predator doesn't do that. Predator gives you information by which you can form a model.
Okay.
And then you can use that model to try and figure out if you're going to make mutations. Where do you put those mutations?
Okay, so you want a rough structure based off, so like in Predator it'll say that 49% of it is alpha helices and stuff
Yeah.
like that, and then we do a rough protein drawing out of that. Okay.
And then quite a bit of it is unstructured, which you'd expect for a protein that responds to tension.
Okay.
So you just want us to like sketch this out on paper and take a picture of it and put it in there kind of a thing?
Yeah, or if you want to draw it out using the Microsoft
Okay.
picture, which is a bit more labor intensive, but that could do it too.
Okay.
Okay, yeah, that was my last question.
All right. Anything else?
I have a quick question about grades for myself. I have most of my grades in there. I just don't have the extra credit for the flu shot or the extra seminar yet.
Well, that's because I don't have an email about it.
I sent an email. It's probably over a month ago by now.
It's not in my emails, which means I didn't get it.
I can find it. I think I sent it to you in an email that had other stuff in the email as well.
Nah, Jenna, it's not there. I've got 3 emails archived from you. One on the 4th, one on the 1st, and another one on the 1st.
It was sent 11/13.
No, it's not. Everything with your name on it is just have those 3. So resend it.
Okay. Maybe I sent it to a wrong email or something.
Well, I just don't get stuff sometimes. Because when I get emails, I archive them, and
Okay. I just like re-forwarded it to you. Hopefully it'll.
okay, don't ask me why stuff like this happens, but it does. Emails they go off into cyberspace, and I just got it. They go
Okay.
off into
I have the same
cyberspace, and
things.
they are never to be found, probably in some undiscovered part of Scotland or something like that.
Right. And I'm sure you get a lot more emails than we do.
I hope to tell you.
I have the same thing, so do you want me to resend those to you as well?
Let me check, Meredith,
Okay.
what I have from you first. Yeah, Meredith, I have one on the 4th, one on the 30th of October, and one on the 18th of November. I don't have the other one. Let's see. Do I? Nope. Nope. And, oh, wait, that's the exam. That's another class.
One was on the 16th.
Yeah, don't have it.
Okay. I'll
Don't
resend
have
it.
it. Resend it.
Yep.
I also sent my flu shot email in from November 20th.
Okay, Greg. Okay, homework number 7. Article summary. Oh, Greg, I'm not seeing it.
All right, I'll send it.
Not seeing it.
I also have the same thing. I sent mine in on the 1st. Well, I sent mine in a while ago, but then I resent it to you, and I have the extra credit for the seminar, but not for the flu shot.
Then resend it. I am literally half the stuff you guys are telling me about. I don't have copies of it. And once I get a copy of it, I send it over the folder for this semester so that in case something happened, there's a record of it there. And that's all I was able to pull out. So just if it's not there, just resend it.
Okay. Do you know if you got all of our like actual assignments then for the last few days that we've been sending you? I think probably I just want to make sure you got like the lab report and.
Yeah, I think I got lab reports. So I have research summaries for Aaron, Greg, Amanda, Jess, Crystal, Andrew, Meredith, Olivia, Selma, Jenna, Divna, Clement, and that's it.
Oh, you didn't get.
I didn't get yours to Dane.
Oh, okay. I sent it, so I'll have to resend that.
Then just resend it. And then I have lab reports for Crystal, Katya, Olivia, Andrew, Selma, Jen, Meredith, Divna, Aaron, Irma, Remington, Greg, Jenna, Amanda, Dane, and yeah, that should be everybody.
Okay, thank you for checking.
They're in the stack.
And then I had a couple of emails throughout the past week. Have you gotten those or no?
Yeah, I got your assignment 7, but it's really hard to read.
Okay, do you want me to try again and make it more clear? Or is it just my handwriting is really bad?
I'll tell you what, can you send me the hard copy through the mail?
Oh, yeah, I can do that.
Yeah, do that. That way, if I'm not sure about something, I can make a note. Then when it comes in, I can just check what I think I read with.
Okay.
That way, I can just check myself.
Okay, so do I just send it to the school and then address
Oh
it
yeah,
to
just
you?
send it to the school.
Okay. Okay, I'll do that then. And then
Yeah.
do my other things look okay? Because
Yeah, everything else is fine. It's just the last exam.
Okay.
Or assignment 7. I forget which one it was, but
Okay.
it was a copy, so it was hard to read.
Oh yeah, for sure. Yeah, I can send that in then.
Yeah. So.
All right. Perfect.
Okay, Dane, I just got it.
Okay, thank you.
Yeah, and Greg, I got your flu shot. And yeah.
Okay, thanks.
And Greg, if there's anything else, make sure that's not, well, not just Greg, all of you. There's something that's not entered, let me know. Now's the time to deal with this, not.
I mean, my seminars aren't in, but that was in a couple of the emails in the past week, so.
And I probably haven't gotten them yet.
Yeah, that's what I figured.
Yeah. Okay, anything else? Nope.
I don't think so.
Okay, guys, if you're on campus, I'm here. I'm not going anywhere, so I'll still come into work, so I'm not hard to find.
Okay, thank you.
Okay.
Thank you. Bye.
Oh, thank
Bye.
you.
Thank
Bye.
you.